Welcome, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Biomodels, I'd like to welcome you to Leveraging New and Established Preclinical Oncology Models for Translational Success. My name is Elizabeth Lamb, and I'll be your host and the moderator for today's event. At this time, I'd like to introduce our presenter for today. He is Dr. Benjamin G. Cuffo, PhD, Principal Scientist, Oncology for Biomodels, LLC. Welcome, Dr. Cuffo. The presenter ball is yours.
Thanks, Elizabeth. I'm very happy to be here this morning to speak with you guys about some strategies for leveraging new and established preclinical oncology models for translational success. Today, we're going to cover a large number of topics, hopefully very quickly and clearly, and then at the end, we can hopefully have time for some questions. First, I'm going to tell you a little bit about Biomodels, which is where I'm coming to you from today. I'm going to give you a quick overview of the standard cancer therapy development pipeline and talk about some of the issues with success rates for novel oncology therapeutics. We're going to talk about a little bit about gradients of predictivity in preclinical oncology models and how we can use rational approaches to leverage these models so that we can make our therapies have the best chances of success. And part of this is the importance of recapitulating clinically relevant context and talk a little bit about novel applications and opportunities in the preclinical oncology space. So first, a little bit about Biomodels. Biomodels is a preclinical contract research organization. We're located in the Metro Boston area, specifically in Watertown, Mass., which is just outside of Cambridge. We were founded in 1997 out of the Brigham and Women's Hospital in Boston. We're now an independent company. Our mission is to design and execute highly translational models of human diseases and conditions in close collaboration with our clients in order to transition drugs and biologics from concepts into patients. And we're happy to report that we've been quite successful with this over the years. We've facilitated almost 50 drugs now to serve patients over a wide spectrum of indications. We carry therapeutic expertise in modeling of oncology, a wide range, as you'll hear some of today. Also, cancer supportive care, treating cancer treatment toxicities, inflammation and autoimmunity, that's a lot of what we do, fibrosis and pulmonary indications. We've just started a great new project with a gnotobiotic facility that allows us to do microbiome studies. We also do cardiovascular disease, metabolic disorders, neurology, and some others that I haven't listed here. So just a quick overview of the standard cancer therapy development pipeline. As you all may be familiar with, we start on the left, and we want to first identify a target. And most of what you see on the left side is all done in vitro. We're going to identify our targets. We're going to identify compounds or biologics that can hit those targets, and we're going to establish our first indications of activity. Following that, we're going to select our clinical candidates and move into full systems, usually rodent systems, to have our initial test of efficacy and safety prior to moving into clinical trials in humans. So Biomodels, for the most part, functions in this realm, but we like to look at this as a full spectrum. I'm going to talk a lot today about how a little bit of forward thinking in the preclinical space can really increase your chances of success once you move it into the clinic. So what you need to get into the clinic and to have an IND that's going to be administered to human patients is basically just evidence of safety and efficacy. So not much is required. You just need to know that the drug will be safe and it has some efficacy. However, success rates for novel oncology therapeutics in particular are very low. And that's not despite a whole bunch of effort that's been put in. So novel oncology treatments that are both safe and effective are continuously in high demand, as we know. And great research that continues is continuing to enhance our understanding of the biology of cancer, and great ideas are continuing to produce innovative treatment strategies. And hopefully you have one of those, if you're listening, under your belt right now. Also, massive investment has been made in the development of novel anti-cancer therapeutics, and people will continue to put money into this because for the patients and also for the opportunity to bring these drugs to market. However, the historical and current success rates of novel cancer treatments remain very, very low, and you can see this table by phase of the probability of approval. And novel anti-cancer therapeutics the success rates are historically lower than even in other indications. So you can look at these graphs on the right and you say, okay, where is oncology falling behind other indications in terms of approval? You say, well, phase 1 to phase 2, everybody's kind of equal, phase 2 to phase 3. But it's really in phase 3 that oncology drops off compared to others. So part of this could be design and execution issues in clinical trials. Obviously, we're dealing with very complicated clinical situations often. We have difficulties often in correctly matching patients to the treatment. And a lot of times we have situations where we're rushing into these clinical trials with only minimal, imperfect, or irreproducible preclinical efficacy modeling. And I'm going to focus on this aspect today. So what are these clinical trial phases, and how can we leverage the preclinical models to enhance our chances of success through the phases? And if you have something even that looks really, really good, how can we gather evidence that will allow us to get expedited review by the FDA and bring our blockbuster treatment to patients even quicker? So one thing that we are seeing more and more, and you can see this graph is from a review done by AstraZeneca in Nature Reviews Drug Discovery in 2014. And you can see that patient selection and stratification is absolutely critical for this. So if you have a high confidence in your patient that will be benefited by these therapies, you can see that 90% of the projects are remaining open with high patient confidence. But if you have a low confidence, most of the projects are closed before they reach fruition. So preclinical studies can be used to help predict patient response if used correctly. Also, the importance of confidence in your target and your mechanism. So this is another way to help stratify patients, but also just know at the basic level that your therapy is engaging your target. So if you look at the graph on the left, you can see that projects that have human genetic linkage of the target to the disease have a little bit of an advantage in success, but projects that have a high confidence of efficacy biomarkers that can stratify patients have a much greater chance of success. And a lot of times what will happen is you want to push forward and you think, okay, well, we'll just figure this out in patients where the situation is a real clinical situation. But if you look in the graph on the right, you can see that as you go through the phases, you don't really establish a greater confidence in your target as you move forward. So you can't expect necessarily to learn more in patients in the clinical phases. There's a number of additional factors that may arise in the clinical setting that should be considered in terms of establishing these biomarkers and finding target engagement. That's, of course, standard of care treatments, what the patients are already getting and how that might interact with your treatment. Their immune status for many of the current therapies to take advantage of the immune system. And to take this one step beyond, the potential effects of the microbiome on immune status and other physiological activities. So the confidence in the target does not increase in clinical trials, as I just mentioned, but we can vet the target mechanism and target engagement in clinically relevant or recapitulative settings in preclinical studies. So what are the phases? Phase 1 is just initial safety testing. We're going to determine a safe dose range in a small number of people. And the primary reason for failure in phase 1 is basically safety, and usually due to off-target effects. So how can you potentially mitigate this in preclinical strategy? You can test safety, efficacy in clinically recapitulative preclinical contexts. You can do dose ranging preclinically and to predict the efficacy, safety windows. You can carry your studies out for longer-term safety studies and look at off-target animal tissues and do some analysis there of potential toxicities. Once we're into phase 2, we're really trying to determine scheduling the patients and identify patient populations most likely to benefit. The primary reasons for failure in phase 2 are efficacy and sometimes patient accrual. So preclinically, or before we go into the clinical trials, or even in parallel, we can work to establish confidence in the mechanism of efficacy using animal models and gathering data. We can use preclinical efficacy models, not just that show any efficacy, but models that specifically are designed to test the mechanism that you think is at work here. And you can vet efficacy in increasingly challenging clinically recapitulative models, not just say, okay, let's set the bar really low and move forward, and then have it fail after you've spent a lot more time and money in patients, but actually really challenge in the preclinical sphere. And you want to test efficacy in context. So getting the context right in a living, intact system is really important. You might want to test multiple contexts and situations. And especially to learn from non-responders to differentiate between if some of your patients or animal avatars are responding and some are not, why are some of them not responding? And really to investigate the non-responders as you would the responders. This will help in patient selection. And to just gather more data, establish non-invasive biomarkers. Anything invasive that requires a biopsy, et cetera, will affect your ability to accrue patients into the study. Establishing non-invasive biomarkers is a great way to increase your chances that you'll move through phase 2. In phase 3, we're really looking at larger groups where we're testing efficacy in clinical context, combinations, comparisons with the standard of care, and it's really the final evaluation of the risk-benefit compared to what's currently available for patients. And the primary reasons for failure at phase 3 are mostly efficacy, but sometimes non-scientific variables that have to do with commercial potential. So how can you try to use preclinical modeling to help through this phase? Well, you want to test your therapy in combination with the standard of care. You want to try to recapitulate in animals the clinical standard of care treatment schedules and ask how might the combination standard of care treatment impact the efficacy of your treatment? How do they work together? And you want to use models that might account for off-target as well as on-target co-toxicities of standard of cares. And also think about how you can benefit treating with the standard of care or in a situation where you've already encountered standard of care resistance, and how does that alter the biology and your target engagement. So at the end of the AstraZeneca review in the Nature Reviews Drug Discovery, they come up with a paradigm that they call the 5 R's of therapeutic development success. So first you need the right target, a strong link between target and disease with differentiated efficacy. You want readily available and predictive biomarkers. You want the right tissue. You want to make sure you have adequate bioavailability and tissue exposure. You want a well-defined PD biomarkers. And you want a really clear understanding of your PK, PDs in the preclinical, moving into clinical. And also understanding drug-drug interactions that are clinically relevant. You want the right safety, of course. You want very clear safety margins, understanding of secondary risks, including metabolites, et cetera. And you want to make sure you're in the right patients. You want to identify your patient population as early as possible and vet this and test this and understand, is this risk-benefit for that population going to be better than what they already have? And that moves into the right commercial potential. So as we look at this overview again, what's required is just safety and efficacy, but what really is going to help and what we recommend is to not only have the safety and efficacy, but have the mechanisms really well defined, including resistance mechanisms, your patient stratification with biomarkers that are hopefully non-invasive if possible, your comparisons with your standard of care, and also, if possible, to get combination timing and schedules down with your standard of care to maximize the treatments to work together and not be working against each other. So just to sum up this introductory part, why invest more of your research and development dollars in preclinical studies prior to or in parallel with clinical trials that you might be able to get into? Well, ultimately, you'll save money and save time. You'll decrease your false negatives and be able to identify patient populations most likely to benefit, and hopefully you'll be able to establish meaningful endpoints for your clinical trials, test therapies in combination as they would be ultimately prescribed, and then increase confidence and thereby increase your success rates overall. So in order to do this, we have a really stacked toolbox that we can use in preclinical oncology, and this is not even the molecular toolbox. This is the more of the animal modeling toolbox, and we can mix and match these to tailor the preclinical studies to look at what you need to look at. And these models are not all created equally. Some of them are more predictive and more accurate than others. But we can, when we use them rationally, we can maximize our accuracy and our chances of predicting response in patients. And these, when we talk about smart models, we consider multiple complex clinical and biological factors. We want to take advantage of known or likely mechanisms of action, utilize specific clinical contexts that are likely to be the case for that indication, recapitulate clinical situations such as surgery, and rest upon translatable endpoints. And often this is done in an iterative fashion. So we keep challenging, we keep looking, and we keep testing. And if something falls off somewhere on the hill we're climbing, well, at least we didn't waste time and money pushing into patients before we had a good idea of what's going on. So I'm just going to go through a few of these quickly. So I'm going to start with genetically engineered mouse models. So these are useful often in defining mechanisms, looking at a number of different things. These are mice that have been engineered in a wide number of ways where we can have spontaneous tumor genesis, which is development at the most physiologically accurate situation, except it's a mouse. But also we can humanize various parts of these animals on many scales and look at human targets in mice. And these mice can be combined with a number of the other models that I'll be talking about. The GEMS models are great for a number of applications, but again, they don't model the particulars of human biology. Cells are grown in accurate context. You can model a lot of things like tumor stroma interactions, tumor immune interactions, and you can do some visualization often by using reporters. Some of the disadvantages of GEM models are they are 100% mouse unless they've been humanized in some way. They tend to be costly because they take a while to develop. You're going to be much faster grafting something into a mouse than developing it de novo, and they're costly because they're time-consuming for the most part. Some of the models are also under IP and need to be licensed. So back to the toolbox, I'm going to tell you quickly about probably the most popular models that people use initially. Those are the subcutaneous tumor models. We combine these with the genetically engineered mouse models. We can apply tumors from mouse to mouse, or human tumors in mouse. That would be syngeneic models, mouse to mouse, or xenogenetic models that are human tumors in mouse. We can combine these with chemotherapies or surgical resection. I'm just going to go through a couple of these really quickly. So your subcutaneous allografts or xenografts are basically just modeling tumor growth in a complete in vivo system in an inaccurate tissue context. So you get a fast and inexpensive way that you can systemically dose your animal and do initial assessments of safety and efficacy windows. Well, we're not modeling as many important physiological aspects of tumor biology, and that's okay sometimes. This is where we would generally start. to get a first look, a first idea of efficacy in vivo. However, you can see at the bottom there are some disadvantages. Because we're growing in inaccurate context, the cells tend to proliferate more rapidly than in context. You're not getting accurate tumor stroma interactions. You have low spontaneous metastasis, which might not be the case in the real situation. And the therapeutic access of your therapy is even not going to be very accurate. The tumor is not in the place it would be. And actually a very low correlation with clinical efficacy, even though the vast majority of the currently available cancer therapies have gone through these models at some point. It's the major way we do it. But you're looking at a small percentage of mice that can be cured from this model that will actually translate into the clinic. So we suggested maybe we can use this as a first pass of more comprehensive studies, looking at initial efficacy following systemic dosing, establishing your efficacious dosages and schedules, and establishing, importantly, your safety FD windows. You can look at safety efficacy in combination with current standard therapies and look at timing and really start to get this nailed down, start to vet your target engagement and get more confidence in your mechanism before you move to these more complex models. I'm just highlighting here the immuno-oncology syngeneic models that can be used. Also, we can use PDX models that have a lot of patient data associated with them in these models as the first pass. We can use GEMS models, and just trying to highlight the combinations that can be used even at this first stage to increase. We can also transduce cell lines or even PDX lines to express bioluminescent or fluorescent reporters, and we can actually run metastasis models with some cell lines that do metastasize in these cases. So below, I'm just showing you a mouse that has a sub-Q tumor, and what we've done here is we've resected the tumor at some point. It's before day 41, that's sort of the middle, and then you can see by day 76, this melanoma has now we can now see it in the brain and possibly spinal cords, CNS, and also in the lungs. So we can use this kind of a model to assess metastasis very elegantly and be able to quantify it with these bioluminescent reporters. So going back to the toolbox, and I'm going to highlight next sort of some of our newer work using germ-free and gnotobiotic models in oncology, specifically syngeneic models in combination with standard care or what will be standard care therapies, and assessing potential therapeutics that modulate the microbiome. So we had the opportunity to try to reproduce this paper that's attracted a lot of interest from last year. And this is a paper that was looking at how commensal acetobacterium can facilitate the efficacy of an anti-PD-L1 immune checkpoint inhibitor. The connection between cancer and the microbiome is our knowledge is growing almost daily, and there's so many different aspects of this, and most of them seem to run directly through the immune system. So it has a definite huge impact on immunoncology and immune-related therapeutics for cancer. But also in terms of treatments with standard of care therapies and others, these can all be impacted by the status of your microbiome or even lack thereof in the case of if patients are receiving antibiotics and their microbiome is altered in that way in terms of being wiped out in some ways. This is a very big burgeoning field, and it seems to have a large role in both new treatments and existing treatments. So this is the study. We compared Jackson mice to Taconic mice, each containing an individual microbiome that is specific to that vendor. And we looked at that in combination with a negative control, the BRAF inhibitor, dabrafenib, in a B16F10 cell line that's transduced to express a synthetic peptide that makes it more visible to the immune system. So B16F10, we know, does not have an activated BRAF, so dabrafenib is our negative control in this case, but does respond to PD-L1, unlike B16F10 without the peptide or B16F0. And then we added just oral gavage of placebo or a combination of both to see how this would affect tumor growth in the sub-Q. And I just added some of the endpoints we collected for this study. We can collect a lot of different things and get a lot of data that may be very well relevant and may provide biomarkers in the future. So in this case, we were just collecting terminal serum via cardiac lead and do a bunch of analysis on that. And just looking at our microbiome composition by collecting fecal contents at various points in the study. Well, that's the terminal, but we collected feces to do that. So just a quick overview of the different handling protocols and the different levels of containment for microbiome studies. Conventional handling, the animals are exposed to anything in the environment. They come from the vendor with their microbiome when they come from, and then as they eat their diet or breathe the air or interact with other animals, they're modulating their microbiome in sort of a haphazard way. If we move up, we have a specific pathogen-free handling where the animals are kept in individually ventilated, HEPA-filtered cages and handled only in pre-sterilized laminar flow hoods. And this really prevents any exchange of microbiome. We can add a sterilized diet and water to this to really contain the microbiome that they come with from the vendor to the microbiome that they have throughout the whole study. Similar is true for gnotobiotic. This is where we have germ-free mice that come to us from Therapeutic, and they are kept in isolators, completely germ-free, but we can repopulate them with specific microbiome of our choosing, either human or murine, and keep that microbiome intact as they live in these germ-free, completely isolated environments. And then the germ-free is the same, only we're not repopulating them with anything. They're just completely germ-free, and this can serve as a model for many different aspects of cancer biology, including modeling antibiotic treatment regimens or activities in terms of the microbiome and immune interface. So going back to the study, we saw that our Jackson tumors grew, were not as great at seeding as in grafting, but once they engrafted, they grew much more rapidly, and the Taconic mice were less, grew less rapidly. And this is an SPF handling protocol where the animals are sticking with the microbiome that they come from the vendor with. When we added dabrafenib, it had no effect as expected, and as negative control, the cell line is not dependent on BRAF. But if we did see an effect there, maybe that would be something to think about as we move forward to the clinic with a potential therapy in here. And then when we add the PD-L1 antibody, we see a really great response as expected. When we added the bifido, we didn't actually see much of an effect in this case, sort of at odds with what the paper had reported. And there's many reasons for negative results, but this is what we got here. So I just wanted to highlight our germ-free gnotobiotic facility. We're up and running, doing a lot of exciting work. So far, we have 14 isolators housing germ-free or gnotobiotic animals, and we've already run a pretty wide range of germ-free and gnotobiotic models over many indications. And it's really interesting the things that people are coming to us with the different models to run in these. So back to the toolbox, I'm going to talk quickly about how we can use orthotopic models. So for the orthotopic models, just to start with the syngeneic, with the mouse in mouse, we can implant murine tumor cells into the corresponding tissue of origin. And these are especially useful when we use bioluminescent and fluorescent reporters in conjunction, and we can thereby monitor the growth of these tumors very accurately at the orthotopic site, the proper tissue contact that we would not be able to look at generally otherwise. So this is great for immunoncology models where we need an intact immune system, and we can do a ton of downstream analysis to establish biomarkers and to vet target engagement, including tumor infiltrating lymphocytes. We have flow cytometric protocols for looking at these. We also can do it by immunohistochemistry. We've started doing some tumor IgG typing to look at tumor antigen acquisition by the immune system. We can, of course, do qPCR, and we can use that to look at a number of readouts, not only gene expression, but biodistribution of, say, luciferase expressing cells within the mouse, rare cells that may be somewhere in the mouse. We can find them with qPCR. We can do cytokine analyses with multiplex ELISAs or standard ELISAs, and more standard CBC analysis. Not going to spend too much time here. So here's just a quick example study of a syngeneic orthotopic model of glioblastoma. We have 3 groups. All were injected intracranially, the GL261 tagged with luciferase, and treated with standard of care temozolomide or fractionated radiation, or the combination. And we can get some nice curves looking at tumor growth and how the cells react in these cases. And of course, on top of that, we can add your novel therapy to see how they would react in these clinically relevant situations. So back to Toolbox. Just quickly, I'm going to talk about some work we did with using PDX models in the orthotopic at the orthotopic site with some clinically relevant standard of cares. So these would be human, in this case, patient derived with well-characterized patient information that are implanted at the corresponding tissue of origin in mice. The mice are, of course, immune compromised, but what we're modeling is basically we're able to model three-dimensional human tumor growth in vivo in contact with a well-characterized patient-derived cells. We can look at the availability of therapy to the primary tumor site, and we're in this case we're able to look at human cancer targets, and we can combine these with humanized murine systems such as hematopoietic, immune, or stroma systems that can help to make these models even more accurate. So quickly, we did a study recently where we tried to model a metastatic breast cancer with adjuvant therapy following resection of the primary tumors you might see in the clinic. So for this, we used NSG mice that are among the most highly immune compromised, and we put in a luciferase expressing PDX line. I can tell you offline about more about how these lines are made, but basically we wait till the tumors reach a size, a threshold based on luminescence at the orthotopic site. The primary tumor is then removed, and then we treat the residual disease as you might in a patient. So we know that these tumors come with a lot of information, so we know that in the patient and in other PDX lines after that, other PDX models using this line after that, we know that this line in the primary tends to be a high responder to capecitabine, but a low responder to docetaxel. However, when we look at the metastatic situation, treating as you might a patient, you can see that we had a response actually with all of these. So treating the primary tumor is not equivalent to treating the metastatic situation. all cases, and if that's the situation that will probably be likely the patients will experience, then we might want to think about testing that before going into the patients. So just to tell you a bit more about these bioluminescent PEXs, we're in collaboration with Genentech, who specializes in generating these, characterizing these PDX lines. They do a ton of molecular profiling, genetic profiling, copy numbers and SNPs and mutations, and transcriptomic profiling, including microRNA expression. They've done a ton of histology and other pathology, and they have a lot of patient treatment response data. So this is the current panel of available bioluminescent PDX lines from Genentech, and the two highlighted are the ones that we've validated in these very sort of complete studies that try to recapitulate what the patient might experience. So back to the toolbox, I'm going to very quickly talk about some of our capabilities for in vitro models. Basically, can do the full spectrum of your molecular preclinical oncology toolbox. We can do your standard proliferation, survival assays, your migration evasion assays. We have a ton of experience in doing a lot of stem cell assays, and we can do these all downstream on or prior to your in vivo, or using tissues that we derive from in vivo studies. We can do a ton of flow cytometry or ELISA, as I've mentioned. This is just showing quickly a matrigel invasion assay, looking at if you have something that might prevent EMT or invasion of your tumor cells. We can use this assay where the cells travel through matrigel towards a chemoattractant, and once they come out the other side, they're stained so that they fluoresce, and we can quantify this nicely on a plate reader. And this is just treating MDA-MD231 cells with either a TGF-beta inhibitor or increasing concentrations of TGF-beta. So I'm going to start to summarize now. Having gone through a few of these, how can we leverage preclinical oncology models for translational success? Well, we want to, at some point prior to getting into patients, we would like to accurately model the biology and really look at efficacy in context, in the tissue context, in the clinical context. We want to be able to have confidence in our target and correlate with the genetics and the epigenetics to stratify patients. We want to take into account tumor heterogeneity and those implications for targets. We want to take into account microenvironment and infiltrates, immune and otherwise. We want to maybe look at resistance and progression, immune contributions, as I mentioned, and really consider the impact of standard of care and other first line. We want to be able to produce clinically meaningful endpoints with our preclinical trials. We want our endpoints to be translatable to clinical trials. We'd like to get more data so we can better establish biomarkers, and in this way provide target confidence that you're really not going to get by running early phase clinical trials, or really any phase clinical trials until too late, basically. And you want to take these and hopefully correlate with clinical observations by doing these in parallel. And as I keep stressing, recapitulate the clinical treatment paradigm. And remember to try to establish early the 5 R's of success: the right target, the right tissue, right safety, right patients, right commercial potential. And just to know that each potential therapy is unique. Everyone is coming up with these really innovative new ideas, and it's important to remember that no single therapy is likely to be effective by itself. So your therapy is going to have to work in concert with standard of care or potentially other new therapies. But preclinical research done well can help identify a therapeutic niche for your therapy. That's why we stress rational and knowledge-based, individualized design preclinical studies tailored specifically to that therapy. In summary, all models are, of course, facsimiles, of course, but the preclinical oncology toolbox can be rationally leveraged to more accurately predict clinical efficacy. The key is how you use the tools at hand. So my case would be that the increased R&D emphasis in the preclinical sphere may probably will ultimately save time and money, and thereby bring better therapies to patients more quickly. Rational use of preclinical models tailored specifically to the therapy indication can enhance the probability of translational success. So with that, I'd just like to say thank you for listening today. Please feel free to get in touch with any more specific questions. And thanks, and have a great rest of the day.